Welcome back. We're up to Lesson 4, What are the building blocks of DNA? In this lesson, students will build a model of DNA, and in that way they will learn a lot about what it is. This lesson, with the next two lessons, go through the central dogma from the DNA to the protein. So it's important that you understand this and read through the next three lessons, this lesson and the next two lessons carefully before working with your students. In the background reading, students will learn that we're at the point where the genetic counselors or the geneticists begin to actually go looking for that specific gene, the HFE gene. Students are going to build a model of this HFE gene starting with nucleotides. This slide here gives us a very straightforward diagram of nucleotides, and students can see clearly that a nucleotide is made up of a sugar, a phosphate, and a nitrogen base. Excellent, easy to understand diagram here. To begin with, let's be sure you're comfortable with what represents what in your connects pieces. If you get directions in your connects pieces, meaning it's fresh from the manufacturer, please throw those directions away as it guides you to in your teacher's edition. Those are not the correct directions. Everything about this model is carefully color-coded. In this lesson, we're going to use the gray and the yellow sugar parts of our nucleotide. There is an orange part, so don't get that confused with the yellow. We'll use the orange part in the next lesson. Each nucleotide will have a sugar base, a phosphate group, which is purple, and one of the four nitrogen bases. For the first strand of DNA, we'll use all gray sugar bases, and for the complement strand, we'll use the yellow. We won't use the hydrogen bonds until we begin building our complement strand. And the hydrogen bonds are color-coded as well. One is double and one is triple. The first thing you'll need to do is build your nucleotides. And a word about that, when you're breaking down your models, you can just leave them at the nucleotide level. Allow students to quickly put together nucleotides before beginning to build the model. We don't want students to just build a model based on one little piece at a time. We want them to understand that these are structures, and into themselves they're structures, and they're part of DNA. So the nucleotides need to be ready in advance, and each group should have their own set of nucleotides. A pair of two students will eventually work with another pair of two students in this lesson. So each group of four students can share a box or bag of nucleotide soup. In this lesson, we will be building a copy of DNA from each of Jack's chromosomes for his HFE gene and each of Jill's. And of course, both Jack and Jill have two chromosomes for the HFE gene. I didn't exactly say that correctly, where the HFE gene resides. And it seems then that we would have four groups of students as we're looking at four models of genes. However, that might take a long time because each of these genes has many parts. And so what we've done is broken down the genes into a part 1 and a part 2. So take a minute to study this and be sure that you understand how this works, and make sure your students do as well. So you'll be building a model of this DNA sequence. A pair of students will build this half, and another pair will build this half. So the gene copy A goes together, but it's being assembled in two parts. Jack's gene copy B is being assembled in two parts, and the same with Jill. For each of these parts, there's a corresponding worksheet. So you'll want to make sure each pair of student gets a worksheet and follow the directions about how to label a model as they build it. That will keep things a lot more simple. Given on each worksheet is the first strand of nucleotides, and they're labeled. Be sure students understand that they're going to build their DNA sequence. They're going to build their DNA strand from the 5' end to the 3' end. How do they know which is which on their nucleotides? Let's look at this adenine nucleotide just here. The dangling phosphate end represents your 5' end. When students go to build their first strand of DNA, they will follow the directions on their worksheet, and they will build from the 5' to the 3'. Again, the dangling phosphate will be pointing to the left to correspond with the worksheet. There's a nice scientific diagram for students to see a little bit more detail what this looks like. Once students have built that first strand, it's time to start talking about the base pairing rules. And now students will write that out before they build their second strand. And we tend to want to write from left to right. And even though this is just on paper, you'll want students to transcribe from the 5' to the 3'. So ask them to start writing their complementary letters, their complementary bases, from right to left this time. And by the way, the reason they're in groups of 3 is to correspond to the codons in later lessons, which will then be used to build a protein, in case they ask. Here is an example of what a double strand of DNA might look like once they've built it. And once again, it's very important that students not just copy something like this in whatever order they want to. They need to build the first strand from the 5' to the 3'. As they build the second strand, we'll look at this next slide. It gives instructions on the order. As they build the second strand, they'll want their hand to represent the enzyme, or an enzyme as it goes out into the body and searches through the bag of nucleotides, what we call the nucleotide soup, finds the correct nucleotide, attaches it with the correct hydrogen bond. Out goes the hand to find the next nucleotide and bring it back, attach, and bond. Search, find, attach, bond. Search, find, attach, bond. Keep the story going the same way that it works in the body. Building this model is modeling. Once your model is built, it's time to put it on the stand. This picture may help you orient this part of your model. It's not real clear in the picture in the book. It looks sort of like a jumble. So you can use this and refer back to it if you need to when you're building the base. All right, so that's the piece that goes right there. And you'll attach a rod to that after putting your legs on. You'll attach a rod. And one student will need to, well, this takes 4 hands. You'll see when you do it. You'll slide your double-stranded DNA down onto the rod, and it's not twisted yet. This time it's not twisted. Be sure your hydrogen bonds are turned so that the hole faces in the correct direction to be slid down onto the rod. Now remember, the rod is not there in real life, nor is the tripod or the quadrupod, just like the Earth doesn't have an axis in it. So be sure students understand that this piece that's holding up the DNA is not part of the actual model. Once you slide this DNA model over this first rod, you can add a connector once you get to the 4th, just past the 4th nucleotide, and you'll have to have somebody twist and push down on the DNA, add another rod, add a connector, and it may take several attempts to do this. Put something on the top when you're done. You should be able to slide your right hand up this DNA model. It is important after you've built your model to ask questions about it, in keeping with science and engineering practices and the cross-cutting concepts of using models. And another type of model is a video, believe it or not. We've included a couple of videos here for you to show students. When it's time to disassemble the model, remember to leave the nucleotides assembled. And yes, you will be rebuilding this model first thing after this lesson when it comes time for the next lesson, but don't let that frustrate you. Students get better at it, and they can take their learning to another level once they've got through building it once. They'll be able to build it really quickly in lesson 5 to move on to the next thing, and they'll also understand it a lot better. Can't wait to see your pictures of your models and to hear your comments, and we will see you back for lesson number 5.